Thank you for joining us on Synthesis Workshop. On today's episode, we're joined by Dr. Benji Thoma. Benji is one of our new resident lecturers, which are specialist speakers that will regularly join us to cover special topics in detail, and the topic that Benji will cover is prebiotic chemistry. To briefly introduce Benji, he completed his undergraduate studies in natural sciences at the University of Cambridge, where he completed summer research projects in the Nietzsche group as well as the Stoltz group at Caltech. He then completed his master's at Cambridge in 2019 in the Gaunt group, investigating activity-based protein profiling warheads. He subsequently completed his PhD in the Powner group, and since then Benji has been continuing his research in prebiotic chemistry as a postdoc. And from there, I'll hand it over to you, Benji. Thanks a lot for joining us today to give this special talk.
Hi, my name's Benji, and I'm a postdoctoral research associate based at University College London in the Powner group. In this video, I'll be giving an overview of the prebiotic chemistry of nucleic acid synthesis. This video is not meant as a comprehensive review of the field, but rather it's an introduction into sections of this field that I think are of interest to synthetic organic chemists. Today we will be briefly outlining the problem of the origin of life, and the organic chemistry involved in it, and then honing in on the structures of nucleic acids. We will take a retrosynthetic approach to thinking about building nucleic acids abiotically, initially focusing on a more traditional disconnection that seeks to synthesize a sugar and a nuclear base, and then bring the two together. Following this overview of the field, I will then go over some more recent advances in prebiotic nucleotide synthesis, mainly the chemistry of amino oxazolines and the stereochemical implications of their chemistry for prebiotic nucleic acid synthesis. Next, we will take a brief foray into prebiotic paths towards alternative informational polymers, such as arabiноnucleic acids, and finally, discuss some methods proposed to enable non-enzymatic nucleic acid extension, that is, how you build an oligomer from RNA or DNA monomers without any enzyme. So, what is prebiotic chemistry? Well, prebiotic chemistry concerns the origin of life. Specifically, we are trying to find out how the first biomolecules formed, and why they look like they do. The astonishing fact is that somewhere on the early Earth, roughly 4 billion years ago, non-living matter became alive. This first cell, or cells, gave rise to all life that is known to us today. So, how were the building blocks of cells synthesized before life existed? This is where organic chemistry enters the scene. But first, we need to more closely examine biological molecules used by life today. Life makes molecules out of carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. Biological molecules are complex, and the manner in which they are synthesized and broken down is even more complicated still. Here, I have taken a short excerpt from the Cake database to highlight the dizzying chemical complexity of life. All of these reactions shown in this slide are catalyzed by an enzyme, which are usually composed of proteins. These proteins are coded for by a sequence of DNA, which is transcribed into RNA before being translated into a protein. This is approximately the central dogma of molecular biology, originally stated by Crick as, once information is passed into a protein, it cannot get out again. We must ask how all of this came about. How were any molecules formed before there were highly evolved enzymes on the scene? In this video, we will be focusing specifically on the prebiotic synthesis of the nucleic acids themselves. Nucleic acids such as RNA and DNA are conserved by all living organisms. Their central role in storing and transmitting information is a central tenet of life itself. Looking at the very specific structures of canonical nucleic acids used by life today is very instructive, as it gives the prebiotic chemist something to aim for. Life uses D-ribose or D-deoxyribose sugars. Ribose or deoxyribose exist in nucleic acids in their furanose form as a 5-membered ring as depicted here. The 3 stereocenters highlighted in blue are set in ribose, and the 4th stereocenter highlighted in orange here is the anomeric center, formed when linear ribose cyclizes to form this 5-membered furanose ring. In canonical nucleic acids, the beta anomer is used, as depicted here. These sugars are used as a scaffold from which further nucleic acid monomers are attached through phosphodiester bonds. These phosphodiester bonds are linked to the 5' alcohol and the 3' alcohol, forming a ribophosphate polymer. At the 1' position of the sugar is the base, of which there are 5 variations. A, T, G, and C in DNA, and A, U, G, and C in RNA. The purines, A and G, base pair with their corresponding pyrimidines. A pairs with either U or T, and G pairs with C through Watson-Crick-Franklin hydrogen bonding interactions. However, at this stage we have to ask ourselves, is there anything special about ribose or about 5' 3' linkages? Can other informational polymers be made out of different but similar backbones? Well, it turns out that the answer is yes. Pioneering work by Albert Eschenmoser has shown that duplex formation through hydrogen bonding interactions is not unique to RNA or DNA. In fact, this property seems to be relatively widespread. Highlighted in this diagram and depicted in boxes are informational polymers made out of non-canonical backbones that can still base pair. So, RNA and DNA were not chosen because of their propensity to base pair and therefore transmit information. We therefore turn to their abiotic synthesis to see whether they were particularly easy to synthesize on the early Earth. If we take an oligomer of RNA and think about how to make it, it may be helpful to do so retrosynthetically. So in the backward sense, asking what bonds do we need to break in order to get back to a starting material that we know. Shown here is a section of RNA with a cytidine in it. We can think about first disconnecting all the phosphodiester bonds, whilst bearing in mind that we need to find a good method of both phosphorylating cytidine and linking those phosphorylated units together. As we shall see, this is far from an easy challenge. Regardless, we can next think about splitting cytidine by disconnecting back D-ribose and cytosine, or C. In order to form the canonical beta anemer used by life, this glycosylation needs to be stereo and regioselective. While it may be simple for a studious organic chemist to achieve this desired glycosylation, in prebiotic chemistry we are limited to what reagents and conditions we can use to those that we think are reasonable on the early Earth. So we're essentially going to be doing synthetic organic chemistry with our hands tied behind our backs. There's additionally the further problem that the version of ribose we want to use for this glycosylation, D-ribofuranose, is in equilibrium with both its linear form and its six-membered pyranose form. So, how can we make ribose, or even better, some sort of version of D-ribofuranose prebiotically? A commonly touted mode of synthesis of ribose is the foremost reaction, which is a relatively old reaction, but one whose complete elucidation of mechanism and pathways continues to engage researchers today. In the foremost reaction, formaldehyde reacts with glycolaldehyde under extremely alkaline conditions that typically employ something like calcium hydroxide. As you can see in this chromatograph depicting the products of a foremost reaction, a wealth of products are formed, and ribose is only a minor constituent of the reaction mixture. However, the presence of ribose has motivated researchers to find conditions under which ribose is selected for in a foremost reaction, and to investigate it further. Now, a true foremost reaction begins with just formaldehyde, which requires on-pole reactivity to form its dimer, glycolaldehyde. As we saw in the previous slide, many foremost reactions actually start from both foremost and trace amounts of glycolaldehyde, which allows entry into this central cycle shown here, which is sometimes called the Breslow cycle. In this cycle, the aldol reaction of glycolaldehyde and formaldehyde yields glyceraldehyde. Tautomerization of glyceraldehyde to dihydroxyacetone and further aldol reaction yields the C4 sugar erythrulose, which can tautomerize to erythrose, its aldol isomer. Erythrose can then fragment through a retroaldol reaction, which gives two molecules of glycolaldehyde. Importantly, this retroaldol step makes the Breslow cycle autocatalytic, as each input molecule of glycolaldehyde ultimately becomes two further molecules of glycolaldehyde, which can then each start their own cycle respectively, which leads to potential exponential growth in glycolaldehyde. Whilst this central autocatalytic cycle has attracted considerable interest, it is idealized because of the wealth of side reactions and off-cycle reactivity that leads to a huge range of products. And this means that this reaction is not directed towards the sorts of sugars we really want to make, like ribose. The huge range of species observed in this reaction are thought to be the result of various aldol reactions, keto-enol tautomerizations, Cannizzaro and retroaldol reactions. despite considerable effort, control over the formose reaction has not been achieved. Some modifications to the formose reaction that researchers have attempted include the use of borate minerals to try and stabilise diols, and therefore ribose itself, or more promisingly using phosphate almost as a prebiotic protecting group in order to limit the chemistry that can occur. For example, Eschremoser has shown that the aldol reaction of glycolaldehyde 3-phosphate and formaldehyde can produce 10 to 20% of ribose 2,4-bisphosphate. However, the phosphate at the 4 position here prevents the formation of the ribofuranose that is used in canonical RNA. Additionally, zinc-proline complexes have been used for a pentose-selective crossed aldol reaction of glycolaldehyde and glyceraldehyde, which can form up to 19% ribose. Moving away from the formose reaction, more recently researchers have begun to rethink prebiotic sugar synthesis. Instead of using aldol reactions to build sugars, the Sutherland group has proposed that the chemistry of hydrogen cyanide and aldehydes can resolve the issues inherent in achieving control over aldol reactions. The addition of cyanide to formaldehyde forms the cyanohydrin glycolonitrile. Reduction of glycolonitrile would form glycolaldehyde, and this bypasses the tricky umpalong of formaldehyde dimerization to glycolaldehyde. In practice, this Kiliani-Fischer homologation of aldehydes can be achieved through thiolysis of the cyanohydrin intermediate, followed by photoreduction of the resulting thioamide. Okay, so we have some ideas about how we can make sugars abiotically, although we will later return to this question and ask whether we need to actually make free ribose in the first place. But thinking back to our original disconnection of nucleic acids, we also need to consider the prebiotic synthesis of the canonical bases, and then consider stitching the sugar and the base together. Now if you look very carefully, the nucleobase adenine, A, is a pentamer of hydrogen cyanide. In the 1960s, it was found that simply by heating a concentrated solution of hydrogen cyanide, very small quantities of A could be obtained. Later work found that the yields of A could be obtained in up to 15% yield in the presence of liquid ammonia, alongside 10% of the species dicyanoimidazole. Painstaking isolation of the intermediates of HCM polymerization revealed the presence of the molecule DAMN, a stable tetramer of HCN, which was found to undergo an unusual photochemical isomerization to yield the heterocycle AICN. AICN itself has remained an exciting prospect in the field of prebiotic chemistry since its discovery as a stable intermediate en route to adenine A. This is because, conceptually, functionalization of AICN with a chloroformamidine unit, or perhaps more plausibly on the early Earth, the simple nitrile cyanamide, has the potential to furnish G. Addition of a formamide unit formally gives adenine. However, both of these pathways to G and A need to selectively hydrolyze the right way. By that I mean, in order to form canonical nucleobases and not bases such as 2,6-diaminopurine, hypoxanthine, or xanthine, we need to be able to control whether a molecule of ammonia or water is lost during the condensation of AICN and either formamide or cyanamide. This is still very much a challenge within the field. Furthermore, even if we could form all of these canonical nucleobases in high yields, would we be able to glycosidate ribose under prebiotic conditions with them? Besides this, what we actually want is a regioselective glycosidation, because in order to form a canonical nucleoside, we need to selectively react with the nitrogen atoms highlighted in blue here. Now, this turns out to be hard, and work by Orgel in the 1970s showed that the purine nucleobases, A and G, were able to form glycosidic linkages with ribose upon heating in the dry state, but they formed the wrong regioisomer. Instead of reacting through the desired N9 nitrogen, they gave the N6 regioisomer. With the pyrimidine bases C and U, no reactivity was observed. This can be rationalized by the fact that these nitrogens in the C and U heterocycles are simply not nucleophilic enough due to their delocalization into the ring. In order to overcome these problems inherent in the poor reactivity of pyrimidines and the undesirable regioselectivity observed for the purines, recent work in the Correll group has looked at synthesizing nucleobase precursors to both purines and pyrimidines, whereby they enact a glycosidation with D-ribose in the dry state with a base precursor that is nucleophilic enough to react. They then unmask the canonical nucleobase in the presence of iron 2 and a thiol. The results from this approach are promising, showing some selectivity for the furanosyl isomers over the pyranosyl isomers. However, the alpha furanosyl isomer is formed in appreciable quantities, and there is still the problem of where this D-ribose actually comes from. The synthesis of D-ribose and the selective reactivity of its furanose form seems like a very difficult problem, and at this point it's instructive to return to our retrosynthesis of RNA to consider whether we might be able to do something different that could bypass ribose altogether. So here again is our molecule of RNA with a cytidine in it. Let's alter our first disconnection slightly this time, not cutting up all of the phosphodiester bonds of the cytidine, but instead disconnecting back to a 2'3' cyclic phosphate. As we shall see shortly, a 2'3' cyclic phosphate may provide a route through to the oligomerization of monomers themselves. And now, instead of thinking of this cyclic phosphate as being composed of a sugar, a base, and a phosphate, we instead seek to synthesize both the sugar and the base on the same scaffold. If we use the formation of the 2'3' phosphate as a means of inverting the stereochemistry at the 2' carbon, then we can see that what we want to disconnect back to is an arabino anhydrocytidine. This anhydronucleotide can be formed from the reaction of arabino aminooxazolene, or AAO, with cyanoacetylene in the presence of phosphate. AAO equilibrates with its ribose stereoisomer RAO in the presence of phosphate. The difference between the arabino and ribose stereochemistry is highlighted with these orange and green markers. In AAO, the anomeric center, shown in orange, is locked as beta because the ring junction has to be cis, and this will ultimately give rise to the natural beta anomer in our final nucleotide product. In RAO, the anomeric center is alpha, again locked in this configuration because of the 5,5 ring system. So the reaction of AAO with cyanoacetylene would give an arabino anhydrocytidine and ultimately the correct beta anomer, whereas the reaction of RAO will give a ribo anhydrocytidine, which would ultimately require inversion at the 1' carbon to afford the natural beta anomer. In order to resolve this problem, we can look to the formation of RAO itself, which is an exciting molecule not only because it can be formed simply from the reaction of ribose and cyanamide, but also because it readily crystallizes. And crystallization is useful because it could have perhaps provided a pathway to separate and purify molecules on the early Earth. Additionally, the crystallization of RAO is made even more exciting by the fact that it crystallizes as conglomerate crystals, which means that a rack solution of RAO crystallizes in distinct L and D domains. This has been the subject of considerable interest with regards to the origin of biological homochirality, and a very recent paper has shown that magnetite surfaces can induce the enantioselective crystallization of RAO. Here, the authors achieved homochiral RAO starting from a solution of RAC RAO. These results were used to infer that biological chirality may be the result of the crystallization of RAC RAO on iron oxide sediments magnetized by the early Earth's magnetic field. However, let's return to our current problem of how we can make RNA from these anhydrocytidines. So, in the forward sense, getting from an anhydrocytidine to a 2'3' phosphate, it was found that a dry state phosphorylation that uses ammonium phosphate and urea afforded the cytidine 2'3' cyclic phosphate in 32% yield. This reaction works because the attack of the phosphate on the 3' alcohol into the 2' carbon of the anhydronucleotide inverts the stereochemistry at this center and provides the correct stereochemistry for a canonical nucleotide. Furthermore, simply shining light on cytidine 2'3' cyclic phosphate leads to the recovery of the starting material in 43% yield, alongside 42% U formed from hydrolysis of C. In principle then, what we have here is an efficient route to both U and C nucleotides, if we can access these aminooxazolines, either RAO or AAO. So what we want is a method that makes RAO and AAO without resorting to making ribose itself. Well, happily, in 2009, it was found that ribose can be completely bypassed in this route through the simple reaction of cyanamide and glycolaldehyde in the presence of phosphate, which functions as a general acid general base catalyst. This reaction pathway, often referred to as the Powner-Sutherland pathway, forms 2-amino-oxazolene, 2-AO, in excellent yields, and 2-AO can then react further with glyceraldehyde to form both AAO and RAO. You'll notice here that in order to form AAO and RAO, cyanamide has to first react with the sugar glycolaldehyde, followed by the addition of a different sugar, glyceraldehyde, to the resulting 2-AO in order to form the amino-oxazolene. This seems to present a problem, as it seems likely that the chemistry capable of producing glyceraldehyde will also produce glycolaldehyde, as in the foremost reaction discussed earlier. And this means that both glycolaldehyde and glyceraldehyde are likely to be present in the same environments. In 2017, a paper by the Powner group showed that these two sugars can be separated through a time-resolved crystallization, enabled by 2-aminothiazole, or 2-AT. In a mixture of glyceraldehyde and glycolaldehyde, crystallization of glycolaldehyde as its aminal occurs first, enabling removal of glyceraldehyde. Reaction of glycolaldehyde with cyanamide to form 2-AO can then occur, followed by the reaction of 2-AO with glyceraldehyde after redissolution of its crystalline aminal. Now, if we take a closer look at the reaction of glyceraldehyde and 2-AO, we can see that 3 stereocenters are set in this one step, and the chirality of the input glyceraldehyde determines the chirality of the resultant amino-oxazolene. This is very appealing because it offers an opportunity to set the stereochemistry of ribose without actually making a free ribose unit itself. There are 4 possible diastereomers formed from this reaction: the desired arabino and ribo, and the undesired lyxo and xylo. The arabino and ribo diastereomers are formed as the major products, and owing to the selective crystallization of riboamino-oxazolene, RAO, that we mentioned earlier, there exists a method of physically separating the 2 major products RAO and AAO. The Blackman group has used this sort of chemistry to achieve an enantioselective synthesis of the arabino and riboamino-oxazolenes, starting from racemic glyceraldehyde through the use of enantiopure amino acids, such as L-proline. In this 2011 paper, the amino acid proline sequesters L-glyceraldehyde, leaving behind D-glyceraldehyde to form its respective amino-oxazolene, further elaboration of which can yield canonical nucleotides. Whilst the Powner-Sutherland pathway to 2'-3' cyclic nucleotides employs AAO, in order to achieve the correct beta anomer as the final product, more recent work by Sutherland has sought to use RAO, which has this attractive property of conglomerate crystallization, in order to synthesize pyrimidine and purine nucleosides. In essence, they found that riboanhydrocytidine could react with sulfur nucleophiles, yielding either thiocytidine or thioanhydropurines. The thioanhydropurine formed is arabino, but importantly, there is a labile C-S bond at C2'. This means that reduction of this thioanhydropurine would afford a deoxyadenosine in its natural form as the beta anomer. The authors found that photochemical reduction of the thioanhydropurine in the presence of bisulfite could do just this. However, the thiocytidine formed from the thiolysis of riboanhydrocytidine is the alpha anomer. So in order to form the natural nucleoside, we need a method of isomerization at the 1' carbon. Whilst this anomerization is hard with the natural nucleobase, it was found that the thione enables this anomerization to the beta anomer in the presence of 254 nanometer light. Whilst almost all of the above work has focused on the synthesis of canonical nucleotides and nucleosides, various chemical investigations have been made into whether simple prebiotic reactions could have also formed other informational polymers that could possibly have preceded RNA or DNA. For example, by exchanging cyanamide for thiocyanic acid, the simple heterocycle 2-thioxazole, 2-TO, can form in good yields. The reaction of 2-TO with glyceraldehyde, analogously to the reaction of 2-AI with glyceraldehyde, yields an intermediate pentose oxazolinone thione, which provides a means for further functionalization towards purine nucleotides. Reaction of this intermediate thione with the trimer of HCN followed by incubation in formamide, gives an anhydropurine, which can be phosphorylated in the dry state to give 8-oxoadenosines. These beta-ribo 8-oxonucleotides are capable of base pairing with pyrimidines, and the oxidation level of these 8-oxopurines allows their assembly as their ribonucleotides through the same strategy as canonical pyrimidine ribonucleotides, as shown earlier. Follow-up work to these pathways has further investigated the chemistry of arabino intermediates, phosphorylation of which avoids ribonucleotides, but hydrolysis gives the arabino monomers. Hydrolysis of arabino anhydrocytidine yields beta-arabinocytosine, but arabino anhydroadenosine is resistant to hydrolysis. Because arabino monomers are still capable of forming genetic polymers that can base pair with a complementary strand, these monomers may still be relevant to the origin of coding and the origin of life itself. Whilst the beta-arabino 8-oxopurines could not be accessed through simple hydrolysis, the use of hydrosulfide as a nucleophile enables thiolysis of arabino anhydroadenosine, which now forms an 8-thiopurine which can be reduced to beta-arabinoadenosine through irradiation. Chemical investigations have been extended to study aminonucleosides, which are prebiotically interesting because of the increased reactivity of their amine compared to the reactivity of an alcohol in water. Recent work has considered the intersection of prebiotic peptide chemistry and nucleotide chemistry by investigating how the presence of an aminonitrile, which is an amino acid precursor, formed through the Strecker reaction of an aldehyde, hydrogen cyanide and ammonia, influences the reaction of glycolaldehyde and cyanamide. The three-component reaction of cyanamide, glycolaldehyde and an aminonitrile proceeds via an intermediate with a 3' aminonitrile, which undergoes a retro-Strecker reaction to yield the 3O and erythro 3' amino-amino-oxazoline. Crystallization of the 3O amino-oxazoline leads to its self-purification, as the erythro amino-oxazoline left in the solution phase is destroyed by cyclization to a bridged bicyclic guanidine. Further elaboration of the 3O 3' amino-oxazoline through the addition of cyanoacetylene and H2S enables the formation of a 2-thiocytidine moiety, photochemical anomerization of which is required to convert the alpha to the beta anomer. Now, nucleoside phosphorylation in water is an extremely challenging problem, but here it was found that the enhanced reactivity of the 3' amino group relative to a diol enable phosphorylation in water in the presence of cyclic trimetaphosphate. Whilst these 2-thiopyrimidine bases themselves are capable of base pairing and are even found in tRNA, it was additionally found that the introduction of these 2-thiopyrimidines to a sulfur-activating species, such as peroxide, can yield the canonical cytosine and uracil bases. Importantly, these 3O nucleic acids are capable of genetic copying, and in particular, aminonucleotides have been used to overcome some issues to do with the non-enzymatic replication of RNA. This is the final disconnection that we need to consider: how we link these monomers together to form nucleic acid polymers. 2' 3' cyclic phosphates have been proposed as plausible monomers to build RNA oligomers from. Early work showed that heating these cyclic phosphates in the dry state in the presence of catalysts such as imidazole, urea, or ethylene diamine yielded small amounts of oligomers. Recently, investigations into the physical setting where this could take place has shown that wet-dry cycling at the air-water interface in model rock pores can form up to 3.5% of oligomerized product without any catalyst present. Another approach to RNA oligomerization is that of non-enzymatic primer extension. Here, you take a short piece of RNA, a primer, which is complementary to part of a longer sequence. If activated nucleotides are added to this mixture, non-enzymatic copying of a sequence can occur. Recent work in the Shostak group has shown that the presence of 2-amino-imidazole, 2AI, can greatly increase the yields of RNA extension through the formation of a 5' imidazolium bridge species. This activated dinucleotide can then react with the diol at the 3' end of a primer in a sequence-dependent manner, forming an RNA oligomer with length +1, with a new 3' end, which another bridged imidazolium can react with. This can occur in excellent yields, but is currently limited to a copying limit of roughly +7 nucleotides. Using an alternative genetic polymer with a 3' amino group on the sugar, just like the prebiotic synthesis of 3' amino TNA we just outlined, leads to both enhanced rates and more extensive primer extension, leaving open the possibility that alternative genetic polymers may have played a role in the origin of life and paved the way for our current informational polymers, RNA and DNA. So, as a quick recap over what we've been through in this whistle-stop tour of prebiotic nucleotide synthesis, I've tried to concentrate on subject matters that may be of interest to the synthetic organic chemist, and as such this video is not comprehensive and necessarily omits lots of great work out there in the field. However, what we have outlined are two distinct disconnections of nucleic acids, one that seeks to synthesize ribose and a base, and then put them together, and the other one that makes ribose and the base on the same molecule. We have also looked at how researchers have sought to make sugars such as ribose and nucleobases, and how they have glycosydated ribose with these bases. We've also covered some of the chemistry of amino-oxazolines, including their synthesis en route to 2', 3' cyclic phosphate pyrimidines, deoxypurines, and RNA pyrimidines. Additionally, we have probed some of these pathways further and seen that alternative genetic polymers such as 8-oxopurines, arabinopurines, and 3' amino TNA are not only possible, but that their functions may have been different to that of RNA and enabled the transition from non-life to life. Finally, we briefly covered how some researchers are investigating non-enzymatic RNA extension and polymerization in order to stitch together these nucleic acid monomers that we have been so interested in making. Thank you for listening. If you're an organic chemist interested in this sort of chemistry, I would highly encourage you to get reading, do some experiments, and get involved in the field.
Thank you for tuning in for this episode, and thank you to Benji for a very interesting and unique talk. If you enjoyed the episode, you can support us by subscribing and telling your peers about this podcast, and feel free to send us any questions or comments you have. Follow us on Twitter to stay up to date, and see you all next time.